It is my great pleasure today to introduce Dr. Debra Bangasser for an interview here for our Psychopharm Friday series. Dr. Bangasser is a professor in the Neuroscience Institute at Georgia State University, and she's also a distinguished investigator of the Georgia Research Alliance and associate director of the Center for Behavioral Neuroscience at Georgia State. Welcome, Dr. Bangasser.
It's lovely to be here, Steve.
Great. And so I thought today we would start our conversation by with your recent paper in psychopharmacology, Williams et al. It's entitled "Early Resource Scarcity Alters Motivation for Natural Rewards in a Sex and Reinforcer Dependent Manner." Could you tell us a little bit about what are the main findings of this paper?
Yeah, so this work was really spearheaded by a very talented former postdoc in the lab, Alexia Williams. And the lab for, I don't know, maybe 5 or 6 years now has been using a model of early resource scarcity, the limited bedding and nesting model. In our implementation in Long Evans rats, we put dams and pups in a low resource environment. There's a metal grate to prevent access to bedding, no enrichment, and only a paper towel for nesting materials. And the dam and pups are in this environment from postnatal day 2 to pups postnatal day 9. On postnatal day 10, everyone's in control housing until adulthood. And Tally Barham started really popularizing the model, and we were very inspired by her work. And she and others, and including my lab, have really focused on how this model affects motivated behavior as it relates to drugs of abuse. But we were really curious about how this model affects motivation for natural rewards, sucrose, and social. So that was sort of like the broad question, like, what does this model do to natural rewards? But we got kind of into the question about like natural rewards based on some other data we had. So when we first started implementing the model in our lab, we implement things a little bit different. Each lab kind of has their own way of doing it. We use Long Evans, which aren't typically used in the model. We have a metal grate as a barrier, so it can go through the cage wash. And we also breed our animals in-house to kind of avoid any potential prenatal shipping related stress. And so we wanted to see whether in our hands-on model changed maternal behavior, which is one of the hallmarks of the model. And so we were looking at changes in maternal behavior, and we do see what I think is consistent with a lot in the field. The moms don't have enough resources to kind of comfortably make a nest and leave the pups on it. So they're often moving around a lot, trying to gather the pups, trying to parent well under these rough circumstances. But we tend to see a little bit more nursing and grooming of the pups at the expense of what we maybe anthropomorphized and called self-care behaviors in the dams. So they rarely rest away from the pups. And they rarely self-groom. And I think it's because they can't just like leave the pups in a nice spot. So we think they have this hyperarousal phenotype. And as we were also characterizing maternal behavior, my lab is really interested in changes in steroid hormones. And so we looked at in adult animals, changes in corticosterone. We didn't see much, but we also were interested in changes in estradiol and testosterone, as we know that stress early in development can change levels of steroid hormones. So we were looking at changes in plasma in adult animals exposed to this model. And we actually found that estradiol levels were higher, um, particularly driven by the males. And so for those of you that don't study steroid hormones, I want to remind you that like estradiol is not just for women. On average, females have higher levels of estradiol than males, but males produce estradiol, and we were really perplexed, like, what does estradiol do in males? And so when we started looking at the literature, we were a little bit surprised to find that one of the things estradiol is really important for is male reproductive behavior. It's actually involved in everything from spermatogenesis to motivation. And so the study that kind of sparked the work in the psychopharm paper is that I had a former graduate student, Samantha Eck, and master student, Jamie Palmer, who were really interested in whether LBN altered male reproductive behavior. And we found subtle effects. So the way you run the male reproductive behavior assay typically is you pair a male with a hormonally primed female. And you actually do this on 3 occasions with a different female each time. And when our lab was learning the assay, we were wondering why. And then we did the assay, and the reason why is like males get better over time, which is good for everyone involved in the assay. But what we found is actually on day 1 and day 3, the LBN exposed males and the control male. So again, LBN, limited bedding and nesting, briefly early in development, now adults. The LBN exposed males perform very similarly to the control males on the first interaction and the second interaction. But on that second day, the LBN males performed as well as most animals perform on day 3. So our interpretation is that they acquired these behaviors more rapidly. And one of the measures where we saw an effect was on latency to mount and latency to intermit, which is the insertion of the penis. And those are thought to reflect kind of the motivational aspects of the task. We also saw a change in latency to ejaculate, which is thought to be more the consumatory behavior. But we had these data and we were like, maybe there's a change in motivation, social motivation due to LBN. Now, this was in a very specific reproductive context, but we were always like, that idea was kind of simmering in the back of our brains. But we really didn't have a great way to kind of test this motivation question in a very kind of controlled, trying to, as best as we can, separate the motivational drive from the consumatory aspects, until we came across Marco Veniero's work. And he's actually an author on this paper, and he developed these social operant chambers where rats can lever press, and then the lever pressing allows for a door to open, and there's a little barrier, and they can interact with another rat on the other side of the operant chamber. And so you can now get, instead of kind of a social interaction task or a reproductive behavior task where it's hard to parse the motivational component, now we have lever pressing and we have progressive ratio. So for those who may not know, on a progressive ratio schedule, rats have to keep increasing the number of lever presses to get one reinforcer. In this case, it's that interaction with the social target animal. And no matter how much that rat likes its buddy, at some point, it's just gonna like give up, and that's the break point. And we tend to interpret a higher break point as more social motivation. So we saw that Marco had this task. And another thing for me who hates building and crafting is that you can buy these boxes now for med associates. So I was like, phew, I can just buy them. I don't have to craft them. They take a long time to come in, but
It cost a
we
lot
were,
too, by the way.
and they cost a lot, but we were so excited about social reward now in the lab. And so we moved. So now I'm finally setting up the psychofarm paper. We had this hint that there might be increased social motivation in males for a hormonally primed female. We have this gap about how LBN affects sort of natural rewards. So in this paper, Alexia, with the help of Marco and another collaborator of ours, Matt Wimmer, really tested whether animals exposed to LBN early in life, postnatal day 2 through 9, now tested in adulthood, have differences in motivation for a social reward and a sucrose reward. And the social reward, we have a lot of questions about the nature of the social target animal, but we started with what we thought was maybe like the most straightforward target, which was a same sex, same age target animal that has grown up in control conditions and is not a cage mate. And what we found is that the males, and this is kind of what we predicted, which is also strange for our lab because we usually find the opposite of what we predict. But in this case, we actually predicted something and it came out okay, where the male LBN exposed rats lever press more on a fixed ratio one lever press per one reward schedule for that social target. And then when we put them on a progressive ratio, they also have a higher break point. So together, we think that the LBN model is increasing adult social motivation in males. My lab almost always includes females in our studies and sometimes only looks at females, depending on our question, if it's about kind of a reproductively special time in the female lifespan. And so we also looked at this behavior in LBN and control exposed females and didn't see an effect on social motivation due to LBN in females.
For another same-sex or
Same sex, same
female.
sex, same age female. We don't know about other types of target animals. And we don't know, for example, if we had a male and she was in estrus, the receptive phase of the cycle, if that would be different. We're trying to unpack some of these questions now because we're really curious about it, but don't have any data yet.
Yeah, it's
But
a harder experiment for sure, but
are always recruiting people if anyone wants to join and do those studies.
oh,
So, so that was social. Now, what about another natural reward? And we chose sucrose. And the way we did these studies, we did not food restrict our animals because we really wanted to kind of separate motivation for sucrose from caloric drive.
right.
And so what we found with sucrose is a little bit similar to the social reinforcer in males. We didn't see much on the FR1 schedule in terms of difference in LBN versus control males, but on the progressive ratio, the males had LBN exposed males had a higher break point, suggesting they're also more motivated for sucrose. So this might be kind of a greater kind of motivation for natural reward that's emerging in the males following this LBN condition. We also looked at females, and I should say that female rats, Long Evans, I think it's true for Sprague-Dawleys as well, love sucrose.
Sure.
It's true for this female human as well.
And this
And so,
male one as well.
so they actually will lever press more for sucrose than the males and have a higher break point than males when they're in the control conditions. But here is where we see a difference emerge in our females where they actually, following LBN, have a lower break point for sucrose. So the males have this like heightened motivation for sucrose and social reward, whereas the females, we didn't see much with social, but with sucrose, they were a little bit less motivated. And now we're planning, we haven't even gotten started, but we're planning to try to do some studies to unpack a little bit more if maybe this effect in females is reflecting aspects of anhedonia. We're not sure. Didn't seem to be there for the social reinforcer, so it is reinforcer specific. And so those I think are the sort of the main findings from that paper.
And then in terms of the social aspect of it, what was the housing condition of the rats? Like, were they solo housed?
This is a really good question. And I think I have to say, I'm going to pepper in like my thoughts about things
Great,
as we chat. As the more I am in the field, the longer my method sections get. Now, with journal word limits, sometimes all those beautiful methods get relegated to supplemental. So please check them out. We are trying to write them as thoroughly as possible because we have made some assumptions that people would think we do things certain ways when we weren't delineating it before. So a question that we got that we started to add to our methods is we do house our LBN exposed animals with their litter mates. So even after the model and after they're weaned, LBN are housed with LBN and controls are housed with controls, and we pair house. Sometimes when they're adolescents, we triple house, but so we never do, even with our drug self-administration studies, they're never socially isolated. So same with this experiment, they were not socially isolated. Now I'm sure you could push the social motivation if we did socially isolate them. And that's a layer of the experiment that we haven't done. That's why it was also, we gave a lot of thought to that initial target stimulus. Sometimes people use a cage mate, but we were a little iffy about that because our cage mate is not the same kind of life history experience because the LBNs are housed with LBNs and controls are housed with controls. So we got a completely novel control raised animal. But I think the results may be different depending on some of these really key variables. Are they isolated with that increased motivation? Do LBN animals interact with an LBN exposed animals differently than a control animal? Like we haven't done all the studies, but there's a lot we could probe there.
Oh yeah, it gets really complex really
Yeah.
fast. And it's but I think these kinds of things are ultimately pretty important because as you just pointed out, like, the details matter a lot, it seems, especially in these kinds of complex experiments where there's lots of different variables ranging from the early life adversity itself all the way up to the social encounter, for example. One might imagine, for example, that a male rat could approach and be interested in sort of self-administering contact with a stranger male for a couple different reasons. One is it wants to talk to the male and see who, be friends or whatever. I'm anthropomorphizing, obviously. Another is that it's like, I hate that guy. Like,
Yeah.
let me like beat him up or something. And you could easily imagine that these then are
And
differentially modulated by something like LBN, for example.
exactly. And we did check. We talked to a colleague of ours who studies aggression, and it was a good lens to get feedback on this project from. It was actually Sam Golden, because he was like, well, are they pressing more because they want to like play or because they want to attack? And I was like, these are rats. They're not mice, Sam. Like they're sweet. But we did do a couple of trials with the barrier open so they could interact just to, I mean, they are playing. It would have been quite an effect if we switched a playful interaction to an aggressive interaction, which we did not. They are curious and want to play, even in the LBN exposed condition. But I was glad someone asked us that before we finished working on the project, because it is important. Why they want to see the animal on the other side is important to try to suss out.
Right. Yeah. And this is one of those kinds of things where sex differences can be coming from a lot of different
Hmm.
reasons, of course. And one of them might be that, a male and a female are simply approaching the task in a different way. Like there's
Hmm.
not in this case, but there could be an aggression component in male behavior more than a female, for example. And that's sort of one of the reasons why you might see different brain substrates and
Exactly.
stuff between the sexes. Of course, there's also other reasons, and I'd love to hear your thoughts a little bit more broadly on the role of sex and how it sort of colors our understanding of how the brain works. Why is it important to look at sex? Like, what are the kinds of things that we know and don't know about sex differences in the brain?
Yeah. So I have been including females in my research ever since I started graduate school. I have to say, when I was an undergrad, I was only male rats only.
You can.
Um, but given my age, that's quite unusual to have been including females in research for that long. Um, and I first just want to say that we don't want to overly dichotomize sex. There's probably no perfectly male brain and no perfectly female brain. On average, male brains have more masculine characteristics, and on average, female brains have more feminized characteristics. I think it's really important to include females in our studies because there are some differences. I already mentioned that there's differences on average in levels of steroid hormones. These steroid hormones at different times in development can actually permanently change the brain. This kind of long-term changes in the brain can also occur in females after pregnancy. Um, and then there's also, we are doing a little bit of work in the lab in collaboration with Art Arnold to look at chromosome effects that control sex differences. So, um, females are XX typically, males are XY typically, and there are a different complement of genes on the sex chromosomes that can have some effects. And there's some beautiful work just on neuroimaging of mice where you can look at sex chromosome effects, and some differences in brain size are actually even driven by sex chromosomes. So hormones and sex chromosome effects are important. So I think it's important to study females because if we're trying to understand the breadth of sex and gender in humans, we're not really going to get there by only focusing on males. Um, I think I think I started to get on a tangent and I forget all the questions that you had in
That
there.
was a very broad question. So sorry about that. But I guess one of the things that really strikes me is, when you're thinking about sex in the brain. So, obviously there's these organizational effects of hormones early in development that can cause some of the more dichotomous kinds of sex
Hmm.
differences. For example, to do with sexual behavior
Right.
itself. Like in rats, like there's a lordosis response in females that's just not there in males. It's just kind of a dichotomous, or maybe it's not quite as dichotomous as I'm saying, but largely so. And so that makes sense to me that there's sort of a reason why you would have a major different mechanism in a female and a male brain for this particular sexual interaction type of a thing. But then what puzzles me is that, we didn't set out, maybe perhaps unlike you, my lab didn't set out to study sex differences, is that we simply have to, because we see them all over the place in everything we're studying, stuff that has nothing obviously to do with sexually dichotomous behaviors, for example, and even on things where males and females are very equivalent in their behaviors, various types of reward seeking, et cetera, seem to sometimes have their sex differences and how the brain does that, how
That's,
the brain
yeah,
creates that behavior. Why? It just
that's
blows my
something
mind
we
every
find.
time.
So I'm gonna approach it, I think, if I can stay on track. This is this is the topic where I have so many thoughts. It's hard for me to keep focused on answering your question.
Gotcha.
So why are there so many seemingly differences in the rodent models, and does that really translate to humans? So one, I always am a little nervous about sort of a ethological argument like, well, females have to parent, so their brains need to be this way. It's kind of hard to like, it's easy to come up with these things after the fact. I'm not really sure. As long as it doesn't prevent females and males from reproducing, it can exist, right? so it might be that in some context we need something like estradiol to work on a mechanism in one brain region for a very female specific behavior, but it doesn't stop estradiol working in another brain region on that same, estrogen responsive element because the gene is there and there's not really a purpose. So I don't think we like things necessarily have to have a purpose. But when we're talking about motivated behavior, I guess to me, it's not necessarily surprising that there might be sex differences. And that especially there might be estrous cycle effects. And we didn't pull out a lot of those in our studies, but I don't know that we did we could be more careful. And certainly if we had a male on the other side of that cage, with a female, I would expect a cycle effect. But, if we think about female and their motivation, when they have the estrous cycle, it's different than a menstrual cycle, is that females are only receptive in estrus. So of course something has to be changing in their brain circuitry to make them receptive that one day. So if we expose them to cocaine that day, it may also not be surprising that they take more. So I think like we're when we talk about motivated behavior, the circuits that drive sexual motivation are paralleling the circuits that drive drugs of abuse. And so it may not be surprising that we find more sex differences in that space, not because it matters that male and females take drugs differently, but because it matters that males and females are motivated to reproduce in certain ways that
So,
help
so
the
basically,
survival of the species.
so am I understanding you correctly in that you're saying that the reason why there's sex differences in many cases in these things that don't have overt sex differences in the behavioral outcomes or even the strategies used by animals of different sexes is because there is a sex dependent or hormone dependent process, which is that your brain better be ready to mate when you're infertile a day of the estrous cycle, and then everything else just sort of has to deal with that particular factor. Like just
That's
what?
one idea. Yeah.
Okay.
I
Uh-huh.
would say another, getting to kind of your second point, which maybe you brought up here a little bit, is sometimes the behavior seems very the same. And when you delve molecularly, like we do a lot of omics studies and we see like crazy sex differences. It's really striking. And so I think another thing that's going on is that a lot of us who went down that kind of had this interest in sex differences research were really interested in identifying things that might bias males and females towards different types of pathology. So we were trying to exploit the differences to understand risk and resilience. But I think now, as more people are including females, particularly in molecular work, where I think including sex was even more underappreciated than in behavior, um, there might be a lot of sex differences, and different people have sort of categorized these with different labels. Hurt DeVries, my colleague, has called them convergent sex differences. Sometimes there's an idea of a latent sex difference, but males and females often have to behave the same or have a physiological response the same way, but that's on a backdrop of different levels of steroid hormones and different levels of sex chromosomes. So to get to the same endpoint, there might be different underlying mechanisms. Like, one of the things we found that we need to unpack a little bit more is that when we did some transcriptomics on the orbital frontal cortex, and again, this is LBN exposed animals, like taking tissue as adults, and we often compare, like, how does LBN change gene transcription in males and how does LBN change gene transcription in females, and then we put those differentially expressed genes into pathway analysis. And one of the things we see is that the glutamate receptor pathway is altered by LBN across sex in males and females. But if you look at the genes that cause that pathway to be altered, they're sex specific. And so we have not we have not done the studies to prove that there are convergent mechanisms, but it could be the case. And Catherine Woolley has beautiful work looking at estrogen regulation of the hippocampus, and estrogen increases plasticity in the hippocampus in males and females, but through completely different receptors and signaling cascades. That work is beautiful. I'm not doing it justice. You should look up Catherine Woolley's papers. She's amazing. But I think as we start to dig molecularly, we may find that in order to kind of have, in the Woolley case, estrogen inducing plasticity correctly against the backdrop of these different kind of baseline differences in males and females, we might need different mechanisms.
So what is that? So is there, so we study, I think you and I both study rats mostly, but we're also both interested in humans. And these are very different species, of course. And there's plenty of other species that are different in various ways, especially when it comes to, social behaviors, sexual pairings or not, all these kinds of things vary quite a bit. I wonder if, given the fact that even something like the same species, if you happen to have an XX or an XY, all the stuff that comes from that sort of causes all these different downstream changes. So even when you need to like sort of have a, change in glutamate response due to LBN, for example, you might come about that in two different ways in the two sexes, as you just said. Does that make you nervous? As I guess I'm saying, I am sometimes made nervous about the idea that then the sex differences and all the kind of details of the mechanisms that we're studying in our rats are going to be at all related to the ones that exist in humans? Or is it that, like, even the same species can go about these things in very different mechanistic ways if you happen to be one or another sex? Why would we then assume that the rats are anything related to the humans, which have so many other differences, obviously, and all kinds of behaviors? Maybe that's too big of a question.
No, I mean, I don't have a satisfying answer for you. I am always worried about translation. I think something that we can really glean from the rats, although it may work differently in humans, as you say, is some of these principles. So I think if we're only studying males, we don't even think if we're only studying males to understand a disease that occurs more often in female humans, I don't think, reducing the complexity is going to be better. So I think by including females, while there certainly are different mechanisms in humans, we may glean ideas about how to approach addressing some of those mechanisms differently. And it may be that for the most part, if we're thinking about developing pharmacotherapies, they work fine across sex. There's such a sledgehammer on the system. The nuances that we're finding in like a transcriptomic paper aren't that important. But I think another way we can sort of leverage sex differences is to glean ideas about vulnerability and resilience. And this is like harkening back to my postdoc work with Rita Valentino, but we found that the CRF1 receptor, so corticotrophin releasing factor, my favorite stress neuropeptide. I'm in the CRF camp. We can fight about it if we want. No, I don't really have a
I don't
side,
know if
but
so, sure.
I was trained in the CRF camp. And so and so um it's a receptor for a stress neuropeptide, and we found that it in the locus coeruleus it signals differently in response to CRF, and the receptor traffics differently. And so, is that happening in humans? I would love to know the answer to that. But does that give us a principle about maybe receptors signal differently, and maybe we need to be thinking about signaling cascades when we're developing CRF antagonists and biased agonists? I think that's where you can leverage it. But yes, if
Interesting.
I start to think about how complex rodents are and then how much more complex humans are, it is overwhelming.
Yeah, but it definitely, I totally agree with you that the principles are what we're really looking for. And this is actually, as it's sort of a little bit of a tangent side note, like this is why I think these efforts to do things like the complete connectome or whatever, where you're like mapping every single synapse in a brain is sort of useless because like other than, because those details are going to differ. Even you learn something and that will change. I mean, like that,
Yeah.
even at that level, and certainly when it comes into species particularities and even sex differences and all that kind of stuff, those details are not the important part. It's what they teach us about the
Yeah, so
principles.
that's how
Yeah.
I look at it, but I wish I had a more satisfying answer.
Well,
I guess my answer is studying only males isn't the answer.
yeah,
But things are complex. Rats are not people. We're doing our best, and they're currently a really good strategy to learn about the brain, which is so complex. We know so little about it. I think um, whatever we learn is an advance.
totally. Yeah. And then what do you think about um, like how does this relate to humans? Because as you sort of pointed out, it's probably obvious to anyone listening. Gender is something that we talk about a lot in humans, and it adds a layer of complexity to this. I don't think we know anything about gender in rats. If they have a gender, like they don't tell us about it. But I'm just wondering if that layer of complexity or whatever is the difference between sex and gender in humans, how that kind of plays out in this context. Like, is there a sexual dichotomy in most cases, and then the gender is sort of layered on top of that, or is it, there's less of a dichotomy than we think? It's more of a sort of analog, spectrum kind of a thing, or what do you think?
Oh, that's a good question. So I want to be very careful to not overly dichotomize sex, because we know that that's not a dichotomous variable. In rodents, we tend to dichotomize it. I've also become better at explaining in my methods section how I'm categorizing males and females. Normally it's genitalia, sometimes it's Y chromosome, but like, we try to be better describing it. But even in rats, it's probably not as dichotomous as we make it out to be, but we're really looking at genitalia. And so gender is for sure not dichotomous. It is on a spectrum. And the way I think most people think about it is more of kind of a psychological construct that we don't know that our rats have it or how to ask it. And that does add a lot of complexity when trying to think about how to interpret sex differences in humans. And so I think that's really kind of a task for the people that do human research studies, to find better ways to ask about the breadth of sex and gender and how they factor that into their analyses. I don't know that gender kind of overrides any underlying sex differences. I can imagine it could, but I just don't think I don't think the field, and this is not my field, so I don't mean to insult human subjects researchers, but my reading of that field is that they're often asking about kind of sex assigned at birth, and that's not capturing the the important factors that we need to think about. And there have been some efforts to guide human researchers in doing better work in that regard. And so I think until they can start to parse that, if that's even worth parsing, like it's going to be hard for us to know.
Yeah, we just simply don't have answers to most of this stuff. There's just it's not
Yeah,
bad
I mean,
enough. It's bad enough that we have so little research on even the dichotomy of the gender, just males or females specifically, let alone the fact that we have like it's barely touched to the
yeah. So I
gender
think
and dissociation.
my kind of thinking on this, it was just always evolving as I learned more and talk to different groups that are kind of affected by this work, is that I think in terms of like what I think we should be doing as preclinical researchers is including females, because I don't think we're going to get at the complexity of the human experience by only studying rats of one sex. I think we're not going to get at the complexity of this human experience by only studying rats or only one age. I think we need to include a lot of factors if we're going to understand humans. But I also think I would say my focus has kind of shifted. I include females and I power my studies to look for sex differences should they be there, in case they teach me good principles or give me clues about risk and resilience factors. But I think our lab is sort of also moving in a direction is that, but beyond just including females, there's a huge gap in thinking about how stress affects females across the lifespan. And actually, some of the work that we're doing now is more not just comparing males and females, but thinking about like what is stress at different time points doing to the females. And we've actually become very interested in the moms in the low resource model, because they're parenting in a low resource environment. And so how is that changing their behavior and how is that changing their brain? So I would say that, I think where preclinical researchers can be really helpful is by trying to be inclusive in the sexes and ages and species and lifespans that they assess. So hopefully smarter people than me can pull it all together and help better understand human psychiatric disorders.
Absolutely. And yeah, that's certainly something that I think about a lot as well, is this, especially in the last several years, this issue of like developmental stage being tremendously important. And obviously these early life adversity models are one example of that. In fact, I think the premise of the LBN model that both you and our lab uses is it's a specific week. It's like the first
Mm-hmm.
week, more or less, of life, a little bit after that. And then you actually take away the
Mm-hmm.
verse situation, the resource scarcity. And I always kind of think about, and I understand why those studies are done that way. Obviously, this is the way I'm doing it that way because my collaborator, Taylor Barham, we mentioned, is that's how she does it. And she's a developmental neuroscientist. You are also doing it, which is great because we can now look at some of the things across labs and so on. But it always does occur to me that like that first week of life, I think the premise is that that's like a sensitive period for
Mm-hmm.
reward circuit development and other kinds of maybe stress circuit development. And then so you're kind of, as a developmental person, you really want to hone in on that sensitive period and see things and demonstrate that things are occurring because they're in that period and maybe not some other period. But also in humans, or at least in, resource scarcity probably doesn't just go away after the whatever first year of life or whatever the analogous period would be in humans. And I kind of always think about or wonder about like to what extent that matters. Like, these animals maybe are, moving from an adverse situation. I'm talking about the pups right now, the young
Yeah,
animals.
right.
They're moving from an adverse situation into now a not adverse situation. I wonder if that transition might be something that's
Yeah,
important as
and I would
well.
also say if you compare models across labs, like my lab uses a lot of enrichment in our home cage. So
Mhm.
relative to other labs, when they move into that control housing, they're moving into a slightly like enriched environment relative to other
Absolutely.
labs. Um, and Mandy Kentner has done some work looking at how a really enriched environment might mitigate some early life adversity effects. It's very cool. You should look it up if you haven't seen it, the audience. So yeah, I think about that a lot. And one of the things we've toyed around with but haven't done yet, but are curious, is trying to extend the model at different time points. And I was just talking with some colleagues who do human stress studies, and we were kind of talking about like what can like, because I'm always bugging them, like, what are you seeing? How can I do my models better? Like, what endpoints should I be looking at? Like, I'm always bugging them. And finally, during our call, I was like, how how am I helping you? Like, what can I do like for you? Kind of, because I'm always asking them for
Yeah.
help understanding things and designing things. And they're like, we really want to know when to intervene.
Ah,
Like, we can't have an intervention that goes all through development. Like, we really want to, like, they also like, and you can look at brain stuff we can't look at, they're neuroscientists as well, but like we really like that would be really helpful. So I did adopt Tally's time window for those reasons. I'm really interested in the development of stress circuitry and reward circuitry, and I wanted to be able to do parallel studies across labs. Um, but I do think there's a benefit to, for those of us that are kind of curious in these questions, to thinking about, are there other critical periods? What does it mean to be in that situation for a longer period of time? And we've toyed around with the ideas and should be doing some of those studies, but we're always behind on what I
yeah,
want to do.
there's always too many questions, and it's a kind of a problem because, like, as you just pointed out, like, so we both use this ostensibly the same model, and we both clearly find major sex differences in some of the same sorts of
Hmm.
behaviors, and yet they're different in some ways too.
Hmm.
And you've mentioned already several of the specific differences that between the exact studies, and the problem then becomes which ones matter, which ones are
Yeah.
causing these changes, and that's the kind of thing that is just really hard to practically study. How do you write a grant on that and get anybody excited
I
about these cleanup type experiments? But
Exactly.
you know,
I don't know the answer. I would love, this is where I'm always like, let's throw more money at the problem. And then everyone's like, I don't have any money. But I have really good ideas about how NIH should spend money. So if anyone at NIH is listening, please give me a call. But I
well,
would
it's rigor
love,
and reproducibility is
that's
what
what
they
I would say.
might
I
call it,
would
right?
love them to invest in, like, say a supplement. So like you and I get a supplement for our grant to like really compare in the rigor. And I would also really, really love um either the NIH or journals to have more standardized methodology reporting practices,
Yeah,
because because sometimes Steve, you and I find different things. I am always like, what did I do different? Like, what is different? Our, like, at my lab was in Philadelphia for when this work was going on. Our like Philly rats, like, tougher than you like
tougher
cool
than Irvine rats, I
California
would guess.
rats. Like, I'm always like, what is it? And I would love to have the opportunity to drill down and really figure it out.
I couldn't agree more. And there's so many things we could learn from that. I mean, for example, you mentioned like five of them. So we have different strains. Does that matter? If so, that's hugely important for interpreting like a lot of other data, not
And
just
we can
experiments,
leverage the genetics,
Absolutely,
right?
absolutely, 100%, yeah. And also the maternal stress or the stress that occurs during the pregnant mothers that are being shipped to our lab normally in our experiments. And you mentioned you're rearing them in house, so that's another developmental sensitive period, of course, is pregnancy, and so their little prenatal brains are kind of doing stuff at that period, and mom's being stressed out is going to matter there too. So these are interacting variables are totally plausible. Totally plausibly causing like major outcome differences, but like again, we just need to figure this out. It is a tractable problem. It's just not the most exciting
And
new flashy method or whatever. So
it's hard to get funded through a normal grant mechanism.
right,
That's why I would love some sort of rigor and reproducibility like supplement. I think these questions are really important, because again, parsing the factors might allow us to have a better understanding of some principles that we could really capitalize on to think about risk and resilience.
absolutely, yeah. And just to kind of even extend this thought into even broader domains. there's so many other differences that may or may not even be in the method section, as you pointed out. So, for example, what is the light cycle that the animals are on?
Mhm.
Labs do this differently all the time.
Yeah,
And
and we do
what is,
everything. We do all our testing in dark cycle
yeah,
or
yeah. So
completely
we do too,
reverse.
but most, a ton of labs don't,
Don't,
and they
yeah,
can't because
they
they're
can't. Yeah.
neither animal care is, they, et cetera, like the timing of staff and so on. All this kind of stuff can make it so that it's just that's just how our lab does it. It's no choice. And also, like, where did the testing occur compared to where the animals live? Are you in an elevator? Are you, around multiple experimenters? Are there people in and out of the colony room all the time? These aren't factors that are just
We've
very
even
difficult to
started
control.
reporting. Um, we have a colleague who does a lot of hamster social behavior.
Mm,
And when they're, my understanding of the story, I might be having it slightly wrong. So don't quote me on it. But when they were moved to, when the facility wanted to put them on the ventilated caging,
mm.
it disrupted all the social behavior. And so I now report like my caging is not ventilated because the ventilated caging, who knows what that's doing to the olfactory development of
Yep.
of an it's different than not ventilated caging, right? So like, yeah,
It really is. It is an issue, and I totally agree that NIH often will say that rigor, reproducibility is a very important issue. They're very invested talking about this issue, but then to actually, you're right. I think that dedicated mechanisms for hashing out some of this stuff, even if it's not going to excite the study section as much, is better. Is money well spent? Because
yes,
it
I
tells us something
I
about.
I agree. We love NIH. We're so grateful for our funding. Steve and I love you, NIH.
That's right.
But if you ever want
Don't get me
tips,
wrong.
tips on where we would love to see investment, you can email us.
Absolutely. Okay, and then one other thing that so this has been such a fascinating conversation. I have like 10 other things I want to talk to you about, but we're running out of time. So I wanted to make sure that
I tend to be long-winded.
oh, well,
Sorry.
it's me too. Believe me, any listener to this knows. Me too. But I did want to ask you, or just ask you a little bit about this guy right here in my background, this
Your bird
bird, and
brain.
I know you have an interest in birds. I wonder if you could tell me a little bit more about that, even either just as a hobby, or if you have any insights about bird and neuroscience connections, that kind of stuff.
Oh, so I came to birds as a hobby. And I think bird neuroscience is absolutely amazing. And there are some really, really cool studies about plasticity and communication that are so valuable in birds. Plus the Ganander finch that was half male and half female that really helped us think about sex chromosome effects. So there's a huge history of bird work in neuroendocrinology, one of the fields where I was trained. That's very important. But that's not why I got into birds. I wish it was. It would make for a better story. I I'll try to be quick. I started hiking a lot when I moved from Southern California, where I grew up, to New Jersey, where I was in a lab with no windows and realized like I need to get outside. And this comes probably to one of the things that drew me to maybe biology and psychology. In psychology, I was interested in psychological questions, but biology maybe is that I like looking at critters. It's probably why I do behavioral neuroscience and not necessarily molecular neuroscience. I like watching animals behave. And so when I was hiking, I was like, identifying critters. And eventually I was like, I want a camera so I can take pictures of things and then identify them afterwards. And really the things I didn't know were the birds. So after years of hiking, maybe I only started my hobby of bird photography like 8 years ago. I got like a bridge camera that was kind of cheap and had a zoom lens. And then I was like, no, I'm all in. And so since then, I am really into bird photography. I don't have a list. I feel like I quantify so many things in my daily life. I can start listing when I retire and don't have to to do all the quantification I do in my daily job. For me, it's about photographing and getting a good shot. And it's been a really good hobby. It gets me out of the house. It was COVID friendly when we were in the social distancing era. And I usually bring my camera to meetings and try to tag on a day of birding there at a meeting. And so I didn't think being a neuroscientist was nerdy enough. So I said, how can I how can I double down? Bird photography.
Oh, that's hilarious.
But I do like bird behavior. So I think it comes more out of my love of kind of the behavioral ethology, which is a field I'm not trained in, rather than neuroscience, although I do think we need more bird research in neuroscience.
Great. Do you have a favorite bird?
This is always very tricky. So I lived in the Philadelphia area for 16 years. And so one of my favorite birds is a tree swallow. It flies really cool. It has a cool tail, flies like an airplane, and it's beautiful blue with a white belly. But March in Philadelphia was when I was started to doubt all of my life choices moving away from Southern California. I'm like, we had a deal. February is the end of winter. Like, why is there ice? But I would see those tree swallows and I would be like, okay, the weather doesn't feel like it yet, but the birds know spring is coming. So for me, that was a very like, it's a beautiful bird, but it's also like, oh, thank goodness March in Pennsylvania is over. So
That's such a great story. I love that, and it's it also, I came the other direction. I grew up in Chicago, and then now I live in Southern California. And one of the things I've noticed is that the lack of seasons really does change some
yeah,
aspects of life. Like, not obviously they can do stuff in the winter. That's not what I mean. What I mean is that going from sort of bad, like winter, cold, brutal, uncomfortable, to that first day of spring and all the things like birds and such that indicate that is it's just a type of psychological
it
feeling you
is.
don't experience
I
here.
never understood. I thought I understood spring fever when I was a kid because we got spring break and like
Yeah.
no school. So like of course I'm happy. But although I actually really did like school. But I never when I moved to New Jersey the first year, I was like, oh, this is different. So elated. So yeah.
It's fascinating. Wow. Okay. Well, as I said, I would love to keep this conversation going for
Well,
another hour probably, but
we will chat
yeah. Well, yeah, great.
offline.
Absolutely. Well, it was really fantastic, and I really appreciate you making the time, Dr. Bangasser, to have this conversation, and I hope people enjoyed it as much as I did.
Thanks for having me. It was delightful. And submit your articles to PsychoFarm. I'm one of the editors. It's a great journal. All right,
Couldn't
take
agree
care.
more. Bye.
Bye.
Thank you.